For all things health and fitness, this is the Army Gymnastics Podcast.
The Army Gymnastics Podcast.
Discover a fitter, healthier you at armygymnastics.com.
Welcome to The Deep Dive. We're the place you come to unpack the really compelling, cutting-edge topics shaping our world.
That's right. We dig into the science, the society, the big questions.
And today we're delving into something incredibly personal. It's sort of a silent companion to every life lived, isn't it? Every memory made. I'm talking about the universal experience of aging.
That quiet dread maybe of decline, the hope for staying vital. It hits close to home for everyone.
It really does. And, the impact of age-related diseases on ourselves, on people we love, it's profound. For basically all of human history, this has been humanity's challenge, something we just accepted.
And you can see why. Historically, aging just seemed like this fixed, unavoidable slide downwards. But that whole perspective, it's undergoing a massive shift right now. Our research for this dive makes that crystal clear.
Exactly. And that shift is precisely what we're here to explore today. This deep dive is ethics and innovation in anti-aging medicine.
Sounds intriguing. What's the plan?
Our mission, essentially, is to take a whole stack of compelling sources. We've got academic papers, the latest scientific news, you name it, and really unravel the advancements that are changing how we even think about aging.
Okay, so the science bit.
Yes, but crucially, we'll also confront the huge ethical questions these breakthroughs raise. Because if we're actively chasing not just longer lives, but healthier, more vibrant ones, well, that changes things.
It really does. What are the implications?
You're about to find out just how far science has come in tackling this ancient adversary. Okay, let's unpack this.
Where do we start?
To really get a handle on the potential here for slowing, maybe even reversing aging, we have to go small. Right down to the foundations, inside our own cells.
Right, because it's becoming undeniably clear, isn't it? The roots of aging are planted right there, at the cellular level.
That's where the latest research really takes us. Our sources point a big finger at a key player, something scientists call senescent cells.
Ah, yes, the zombie cells. You hear that term a lot.
You do. And honestly, the analogy, it's surprisingly good. These are cells that have basically clocked out. They've lost their ability to divide.
So they're not helping repair tissues anymore.
Huh.
Or regenerate things.
Nope. But, and this is the crucial bit, they haven't died off either. They just hang around.
Yeah.
Linger in our tissues, building up over time.
So they're not just neutral then. They're not just like dead weight taking up space.
That's the key. That's what makes the zombie name stick. They're not dead, but they're definitely not healthy.
Right. They are anything but inert. Instead of just sitting there quietly, senescent cells become actively problematic. You can almost picture them as tiny, malfunctioning factories.
Malfunctioning factories, I like that. Instead of doing their job.
Exactly. Instead of doing their normal, healthy jobs, they start churning out this nasty cocktail of harmful, pro-inflammatory signals. They pump it out into their surroundings. This toxic mix, it has a name, the senescence-associated secretory phenotype. SASP for short. And that is a major driver of what we actually experience as aging, the symptoms, the diseases.
So these zombie cells are actively making things worse, broadcasting these damaging signals, like poisoning the neighborhood.
That's a pretty fair way to put it, yeah. The scientific evidence now really strongly links the buildup of these senescent cells and their harmful SSP secretions to how bad a whole range of common age-related diseases get.
Like what, specifically?
Well, our sources detail their direct role in making conditions like Alzheimer's disease worse. They contribute significantly to atherosclerosis, that's the hardening of the arteries, and they definitely exacerbate type 2 diabetes.
Wow.
And it's not just about those specific diseases either. Their presence, as they accumulate, has this damaging system-wide impact. They actively degrade the healthy tissue around them, which compromises organ function pretty much across the board. It's a systemic issue.
That paints a pretty bleak picture. Okay, if these cells are so bad, we need to understand how they end up like this. How does a perfectly healthy cell turn into a zombie? What's the trigger?
It's a complex response, usually to different kinds of cellular damage or stress. Our sources lay this out meticulously. Essentially, senescence is this powerful, built-in defense mechanism cells have.
A defense mechanism? How so?
Well, think about it. If a cell gets significantly damaged, maybe damage that could lead to cancer, it triggers senescence as a way to protect the organism. It stops dividing, prevents potential runaway growth.
Okay, that makes sense. A safety brake.
Exactly. But the problem is, over time, if these senescent cells aren't cleared out efficiently by our immune system, this protective mechanism flips. It becomes detrimental itself.
Ah, the downside. So what are the main triggers that push a cell into this state?
One of the most famous and really well-studied triggers is telomere shortening.
Telomeres, right. I remember these, the little protective caps on the ends of our chromosomes, the ones that hold our DNA.
Precisely. They're like the plastic tips on shoelaces, but for our DNA. They're non-coding, repetitive bits of sequence at the very ends. They act as buffers, stopping us from losing vital genetic info when cells divide. Here's how it works. At birth, our telomeres are typically pretty long, maybe 8 to 12 kilobases, but every single time a cell divides, a little piece gets lost, roughly 50 to 200 base pairs. Gone.
Why does that happen?
It's because of something called the end replication problem. Basically, the machinery that copies DNA, the DNA polymerase, it just can't quite replicate the very, very ends of our linear chromosomes, so a little bit gets clipped off each time.
So with every division, they get shorter and shorter.
Yeah.
Like a fuse burning down.
That's a great analogy. And think about it, over a lifetime, our tissues are constantly repairing, replacing cells. Billions and billions of cell divisions happen.
Leading to shorter telomeres.
Exactly. Progressive shortening. And eventually, they dwindle down to a critically short length. They can't protect the chromosome ends properly anymore.
And the cell notices this.
Oh, yes. It triggers a big cellular alarm system. It's called the DNA damage response, or DDR. This DDR then switches on specific proteins that act like brakes on the cell cycle. P16INK4A and P21CIP1 are key ones. These proteins basically slam the brakes on cell growth. They cause a stable, irreversible stop to the cell cycle. And that specific type of senescence, the one caused by hitting this division limit, sometimes called the Hayflick limit, that's replicative senescence.
And this isn't just some abstract process happening in a dish. It has real-world effects on our health,
Right.
linking the microscopic to the macroscopic.
Absolutely. The correlation is stark. The more cell divisions over a lifetime, the more telomere erosion and the higher the burden of these senescent cells. And the clinical data, it backs this up strongly.
How so?
Well, shortened telomeres in people aren't just a marker of getting older, they're significantly linked to actual age-related conditions like liver cirrhosis. And quite profoundly, they're associated with increased risk of dying from any cause in people over 60. It's a really compelling link between this basic cell process and how long and healthy our lives are.
Okay, telomere shortening is a big one. What else can push a cell into senescence? I think you mentioned something about genes involved in cell growth.
You're right. That's another critical pathway, oncogene activation and tumor suppressor inactivation.
Oncogenes, those are the cancer genes, right?
Sort of. Oncogenes are mutated forms of normal genes called proto-oncogenes. In their healthy state, these proto-oncogenes are essential. They help regulate cell growth and division. But specific mutations can make them overactive, pushing the cell towards uncontrolled growth, towards cancer.
Got it. And tumor suppressors do the opposite.
Exactly. They're the body's natural defense. Their proteins normally halt cell division or repair DNA damage or even trigger programmed cell death apoptosis if the damage is too bad.
So if those defenses fail.
Right. If tumor suppressor genes get inactivated, maybe through mutations or other changes, they lose their protective function. Also increases cancer risk. Now, here's the fascinating part, kind of counterintuitive at first.
Okay.
When certain oncogenes like RAS, BRAF, AKT become overexpressed, or when crucial tumor suppressors like PTN or NF1 get inactivated, this itself can actually trigger senescence.
Wait, so the very things that could cause cancer can also trigger this cell-stopping state?
Precisely. It's like the cell's own internal safety mechanism kicking in, a last-ditch effort. Instead of becoming fully cancerous, the cell hits the senescence emergency brake, stable growth arrest.
So senescence in this context is actually stopping tumor development.
Yes. It's a powerful, built-in anti-cancer mechanism.
Yeah.
It prevents potentially dangerous cells from multiplying out of control.
That's quite elegant from the cell's point of view. But what about damage from the outside world, environmental factors?
That leads us nicely to the third major trigger, mitochondrial dysfunction and reactive oxygen species, or ROS.
Mitochondria, the powerhouses of the cell.
Yeah.
And ROS, those are damaging molecules.
Highly reactive ones, yes. Things like hydrogen peroxide, superoxide ions. They're actually normal byproducts of metabolism, especially energy production in the mitochondria. Usually our cells have enzymes like superoxide dismutase that neutralize them, keep levels in check.
But if the mitochondria start to fail,
Exactly. If they become dysfunctional, maybe due to damage building up over time, they can start leaking excessive amounts of ROS. This flood of ROS creates oxidative stress.
And that damages things.
Big time. It causes oxidative damage not just to the mitochondrial DNA itself, but to the cell's main DNA in the nucleus and other important molecules. And guess what that widespread DNA damage triggers?
Let me guess. The DNA damage response again? The DDR.
You got it. Just like critically short telomeres do. And once again, that DDR activates those same core pathways, P21CIP1 and P16INK4A, pushing the cell into senescence.
So multiple roads lead to senescence.
They do, and it's crucial to remember it's not just internal problems. Exogenous factors, things from outside matter too. Think prolonged UV radiation from the sun or harmful chemicals in tobacco smoke. They can directly cause ROS formation and DNA damage, nudging cells down that senescence path.
So it really is a combination of things. Internal wear and tear, genetic slip-ups, and external attacks, all conspiring to create these zombie cells.
That's a really good summary. And what's also critical, our sources hammer this home, is that these triggers don't usually act alone. They often converge, they interact, and they end up activating those same key tumor suppressor pathways we keep mentioning. P16INK4ARB and P53P21CIP1.
The master switches for senescence, it sounds like.
They really are central to establishing and maintaining that stable growth arrest that defines senescence. It's a complex, interconnected system, fundamentally protective, but paradoxically, it contributes to this buildup of problematic cells as we age. Understanding these triggers is really the first step if we want to intervene.
Okay, that gives us a really solid understanding of the problem itself.
Sure.
Now, knowing what they are and how they form, the next giant hurdle for scientists has been, well, actually finding them in the body, telling them apart from healthy cells, and then figuring out what to do about them. For a long time, that was a huge bottleneck, wasn't it?
Oh, absolutely. A massive bottleneck. For decades, the main way to spot senescent cells was pretty traditional biology. You'd tag them, use fluorescent labels or biochemical markers.
How did that work?
You'd use a molecule designed to stick to something specific on a senescent cell. Then, under a microscope, it would glow or change color, letting researchers see them.
Sounds straightforward enough on the surface, but I'm guessing practical problems, especially with living cells.
Big practical problems. Our sources point out several major drawbacks. First, it's often messy, slow, very labor-intensive. That really limits how many experiments you can do, how fast you can learn.
Okay, efficiency issues.
Yes, but here's the real kicker. The very act of putting those labels on the cells can change the cells themselves. It can alter the very properties you're trying to study.
Oh, wow. So you're interfering with the subject just by observing it.
Exactly. Imagine trying to study a delicate ecosystem, but your observation method scares away half the animals or changes their behavior. It fundamentally compromises the reliability of your research. It's like trying to measure the temperature of something hot, but your thermometer itself cools it down. A real roadblock.
That is a powerful analogy. So you're not just observing, you're accidentally interfering. So the big question becomes, how do you find these cells without messing them up? Is there a breakthrough for that kind of untouched detection?
Yes. And this is where the innovation just gets incredibly exciting. A team based at Tokyo Metropolitan University came up with a truly groundbreaking solution. It's called frequency modulated dielectrophoresis, or FMDEP for short.
Okay, FMDEP. Sounds very technical. Can you break down how that actually works? What makes it so brilliant?
I'll try. Imagine a single cell floating in a fluid. Now expose that cell to a very precisely controlled electric field that alternates switches back and forth. This field makes the tiny charges inside the cell rearrange themselves. And because the field isn't perfectly uniform, this induced polarization makes the cell actually move. Now, the genius part of FMDEP is watching how the cell's movement changes as you tweak the frequency of that alternating electric field.
Okay, changing the frequency.
Yes. And they found a key point, something called the cutoff frequency.
The cutoff frequency, like finding the right station on a radio dial.
Kind of. It's that specific frequency where the cell's characteristic movement, its little oscillating dance, if you will, changes dramatically. And here's the brilliant discovery. That specific cutoff frequency is measurably different for senescent cells compared to healthy younger cells.
A unique electrical signature.
Exactly. A distinct electrical fingerprint.
And why? What's different inside the senescent cell that makes it react differently to the electricity?
It boils down to fundamental changes in the cell membrane, the outer skin of the cell as it ages and becomes senescent. The lipids, the fatty molecules making up that membrane, they change their composition.
Right.
And those changes directly alter the cell's electrical properties, how ions and charged stuff move across the membrane, and that dictates how the whole cell responds to external electric fields at different frequencies. So just by measuring these unique electrical responses, FMDEP can tell senescent cells apart from non-senescent ones, and it can do it incredibly quickly in just seconds.
That is genuinely ingenious, using the cell's own electrical properties against it, in a way.
Yeah.
The advantages must be huge, right? Not just for research, but thinking ahead.
Oh, they're profound. The method is super fast, it's easy to implement, and critically, it's completely label-free. No added chemicals, no messing with the cell's natural state. It keeps the cell viable, intact.
Which opens up
A whole new toolkit for researchers.
Yeah.
Much faster experiments, pure results because you're not interfering, the ability to do real-time studies on living cells as they change.
Wow.
Plus, it's perfect for high-throughput drug screening. You can rapidly test thousands of compounds to see how they affect senescent cells. That really speeds up discovering new therapies. And because it's label-free, it's super versatile. They tested it on human skin cells, but it has potential for many other cell types.
Here's where it gets really interesting. This isn't just a small step. It feels like a paradigm shift. How we might diagnose aging, personalized treatments. Imagine spotting these problem cells early or tracking therapy in real time without interfering. That's revolutionary.
It really is. This kind of innovative detection fundamentally changes the landscape. It moves us way closer to a future where we can precisely monitor aging right down at the cellular level.
And speaking of diagnostics,
Yeah.
moving from the single cell to the whole body. There's another amazing breakthrough, right?
Yeah.
Using MRI to actually see these zombie cells inside us.
Yes. Shifting focus now from the micro to the macro, there's a truly remarkable new MRI technique. This was developed by researchers collaborating between Stanford Medicine and Northwestern University. And it lets us visualize senescent cells non-invasively, in vivo, inside a living organism.
That's astounding. What specific problem does this solve for, say, patients and doctors?
Think about a condition like osteoarthritis. It involves cartilage breaking down in joints. But until now, there was no way to directly see the senescent cells within that damaged joint tissue without, taking a sample.
A biopsy.
Right. Which is invasive. This lack of non-invasive imaging was a major barrier. How do you develop or use targeted therapies effectively if you can't accurately see where the problem cells are, how many there are, inside a patient's body? This new MRI trick provides that missing visual information.
Okay, so how does it work? Is it a brand new kind of MRI machine?
No, not a new machine, which is part of the beauty. It's a clever modification of a standard MRI contrast agent, something called gadolinium, which is used all the time.
How do they modify it?
They added a special chemical cage around the gadolinium molecule. This cage is designed with incredible precision. It only opens up and releases the gadolinium when it bumps into a specific enzyme called beta-galactosidase.
And why that enzyme?
Because beta-galactosidase is found at much, much higher levels, specifically inside senescent cells. So regular gadolinium lights up everything. But this caged version, it's like a tiny spy. It only opens its cage and lights up the MRI signal when it finds a senescent cell.
Selective illumination.
Yeah.
That's incredible precision. What are the applications? Where is this heading?
It's remarkably versatile. The agent can be injected into the bloodstream for a systemic view, or for localized problems like arthritis, directly into a joint like the knee. They've already done successful studies in pigs, implanting human senescent cells and showing that only those cells lit up on the MRI.
So it works in a living system? What about humans?
That's the next crucial step. Studies using human knee joint specimens, the ones removed during knee replacement surgery, are underway right now. That's bridging the gap towards actual clinical use.
The potential here must be huge, way beyond just arthritis, right? If you can see the senescent cells.
Absolutely. It's critical for identifying which patients have a high burden of these cells and are therefore most likely to actually benefit from senolytic therapies, the drugs that clear them out.
Patient selection. Makes sense.
And imagine tracking treatment. Instead of waiting months, maybe years to see if symptoms improve, which is slow and uncertain, this imaging could potentially show you visually, within weeks, whether the senolytic drug is actually reducing the number of senescent cells in the target tissue.
Wow, that's fast feedback.
Incredibly fast. And the researchers are really optimistic this approach can be adapted to spot senescent cells in many other diseases, where they're implicated various cancers, Alzheimer's, metabolic disorders. The ability to precisely visualize these cells could transform diagnostics across the board for age-related conditions.
So we're getting incredibly sophisticated at finding these zombie cells. Which leads us, naturally, to the next big, exciting question. Now that we can pinpoint them, what can we actually do about them? What therapies are being developed to either get rid of them or stop their harmful effects?
Right. That ability to identify them accurately really unlocks the door for targeted therapies. And broadly speaking, these treatments fall into two main camps, senolytics and xenomorphics.
Okay, let's tackle senolytics first. The name itself, senolytics, sounds like it means breaking down or destroying something old. Is that the idea?
In essence, yes. Senolytics are drugs specifically designed to selectively eliminate senescent cells. They hunt for unique vulnerabilities that these zombie cells have, allowing them to be killed off while, ideally, leaving healthy neighboring cells unharmed.
Who are the pioneers here?
A lot of the pioneering work identifying and characterizing these drugs really comes from Dr. James L. Kirkland and his team at the Mayo Clinic. They've been leaders in this field since around 2015, making some really foundational discoveries.
And what are some actual examples? Drugs that are making headlines or moving through trials?
One of the most prominent examples is actually a combination of two existing compounds, dasatinib and quercetin. Dasatinib is already used as a leukemia drug. Quercetin is a natural flavonoid found in things like apples, onions, berries.
Interesting combination. How well does it work?
In extensive mouse studies, this combo showed remarkable results. It didn't just extend lifespan, but crucially, it significantly improved health span, the period of life lived free from age-related disease.
Health span, not just lifespan. That's key.
Absolutely key. And what's really exciting is that small pilot human trials using this dasatinib plus quercetin combo have already been completed. That really paves the way for larger, more definitive studies in people.
Any other promising candidates?
Another natural one getting a lot of attention is fisetin, also identified by Dr. Kirkland and others. It's another flavonoid found in fruits and veggies. Animal studies with fisetin have consistently shown it reduces the senescent cell burden, improves health, and extends life, even when the treatment starts late in the animal's life.
So these aren't just lab curiosities, they're actually moving towards real-world use.
They are indeed. And researchers like Dr. Judy Campisi, another giant in the aging and senescence field, have been really instrumental in pushing to get these senolytics into formal clinical trials for specific human diseases. Things like osteoarthritis and glaucoma are prime targets.
And the results in animals are compelling.
Very compelling. For osteoarthritis, animal studies consistently show senolytics can actually reverse the disease pathology. They seem to promote genuine cartilage repair and regeneration. That's a huge deal.
If we connect this to the bigger picture, this sounds like a fundamental change in medicine.
It really could be. Instead of just managing symptoms or maybe slowing disease progression, we're talking about potentially addressing a root cause by literally removing the cells driving the problem. It could fundamentally change how we treat dozens of age-related diseases.
A profound shift from management to potential cure or reversal. Okay, what about the other category you mentioned, xenomorphics? How are they different?
Xenomorphics take a different tack. They're still targeting senescent cells, but instead of killing them, they focus on neutralizing their harmful secretions, that SASP cocktail we talked about.
Silencing the bad signals.
Exactly. Xenomorphics work by inhibiting or neutralizing the pro-inflammatory, tissue-damaging actions of those secreted factors. They aim to mitigate the negative effects on surrounding tissues without necessarily eliminating the senescent cell itself. They basically put a muzzle on the zombie cell's harmful broadcasts.
Clever. Are there examples of these being tested?
Yes. One compound, called UBX0101, was investigated for osteoarthritis. Another example is roxithromycin, which is actually a macrolide antibiotic. It showed promise in studies for idiopathic pulmonary fibrosis, that's severe lung disease, apparently by blocking key SCSP components.
Interesting. Any others?
Drugs like rapamycin and ruxolitinib are also showing potential, particularly in conditions like atherosclerosis. They work in part by inhibiting a pathway called MTO. mTOR is a major intracellular signaling hub, and it's known to be a key initiator and regulator of SASP production. So by tweaking mTOR, you can dial down the harmful output from senescent cells.
So multiple ways to potentially tackle the problem, either eliminate the cells or silence their damaging messages.
Hmm.
Are there other types of drugs or interventions in the broader aging research field that are generating buzz?
Oh, yes, the field of geroscience is incredibly vibrant. Beyond directly targeting senescence, the American Federation for Aging Research, AFAHAR, highlights several other really exciting areas.
Like what?
Well, there's metformin. It's a super common, safe drug used for type 2 diabetes. But now it's the focus of a landmark clinical trial called TAME Targeting Aging with metformin led by Dr. Nir Barzilai. The big question is, can metformin actually delay the onset of multiple age-related diseases simultaneously? Not just diabetes, but heart disease, cancer, cognitive decline.
Oh, that would be huge.
Absolutely. Then there's rapamycin, which we mentioned inhibits MTOR. It's being studied extensively for broader anti-aging effects. There's a massive project called the Dog Aging Project, led by Dr. Matt Kaberlen and Dr. Daniel Promislow, looking at whether rapamycin can extend healthy lifespan in pet dogs. The insights for human translation could be enormous.
Dogs as a model for human aging. Fascinating. What about boosting our cell's own machinery, like the mitochondria?
Right. That leads to compounds like alamipretide, also known as SS31. It shows real promise in boosting mitochondrial function, remember, the powerhouses that decline with age. Research by folks like Dr. Peter Rabinovich and Dr. David Marcinick shows clear benefits in old mice. Heart, muscle, brain, kidney function improve. It's even being looked at for Alzheimer's.
Okay.
And then you have the whole category of NAD booster molecules. Researchers like Dr. David Sinclair have championed these. The idea is to restore cellular levels of NAD plus T, which is crucial for tons of cellular processes, but declines as we age. Boosting NAD plus might help rewind some aspects of aging. All these different avenues are converging, really reshaping how we approach aging as a biological process.
This ability to detect and then target senescent cells, either by getting rid of them or shutting them up, sounds incredibly powerful. But let's dig a bit deeper into those harmful secretions themselves, the SASP. You said it's a complex cocktail. What's actually in it, and why is knowing that so important?
It is incredibly complex, and understanding SASP is absolutely key to grasping the full impact of senescent cells. It's basically this pro-inflammatory secretome they release. And it's not just one or two things. It's a dynamic mix of hundreds of different secreted factors.
Hundreds? Like what?
You've got loads of pro-inflammatory cytokines. Those are signaling proteins, things like interleukins, IL-1 alpha, IL-1 beta, IL-6, IL-7, IL-8. The list goes on. Then there are chemokines, which are signals that recruit immune cells to the area, things like MCPs and MIPs.
Okay, cytokines, chemokines.
Plus growth factors that can sometimes promote unwanted cell growth or fibrosis, which is scarring. Also proteases, enzymes that literally chew up and remodel the matrix, the scaffolding around cells, and even tiny packages called extracellular vesicles carrying signals and various metabolites. It's a really sophisticated communication system.
But a system that's often broadcasting damaging messages, creating a toxic local environment?
That's exactly it. But here's what's truly fascinating and still a huge area of research. SASP has a dual nature. It's not always bad.
Really? When is it good?
In certain specific short-term situations. Like during embryonic development, SASP helps get rid of unwanted cells as tissues are sculpted. In adults, it's actually vital for proper wound healing. It calls in immune cells like macrophages to clear debris and helps limit excessive scarring.
Okay, wound healing.
And even in the very early stages of cancer, SASP can sometimes help by recruiting the immune system to nip those precancerous cells in the bud. You can almost think of it like a controlled burn necessary sometimes to clear out old or damaged stuff.
So the problem isn't SASP itself, but when it becomes chronic. When the controlled burn turns into a raging, persistent wildfire.
Exactly. The detrimental roles of SASP really come to the fore when senescent cells pile up and their secretions become chronic long-term. This persistent, low-level drip, drip, drip of SASP contributes massively to what scientists call inflammaging. Yeah, it's this chronic, sterile, meaning not caused by infection, low-grade inflammation throughout the body. And it's now seen as a fundamental driver of almost all age-related diseases. This chronic inflammation can also mess up our immune system, making us worse at fighting infections and clearing out damaged cells.
A vicious cycle.
And what's really insidious is that SASP can even spread senescence to nearby healthy cells. It's called paracrine senescence. So the zombie cells can essentially infect their neighbors, amplifying the damage, like a forest fire jumping to healthy trees.
This raises an important question, then, for developing therapies. If SASP is sometimes good and sometimes bad, how on earth do you target it effectively? How do you suppress the bad stuff without messing up the good stuff? That sounds incredibly tricky.
It is, without a doubt, one of the biggest challenges. The key has to be understanding the precise mix of CSP factors in different tissues at different times and how they're regulated. The goal probably isn't to wipe out all SASP, but maybe to selectively neutralize the worst offenders, the really pro-inflammatory, pro-fibrotic ones, while leaving the beneficial parts like immune recruitment for cleanup intact. It requires incredibly nuanced, context-dependent treatments, a very active research area.
Okay, let's connect this cellular senescence and especially this chronic SACP to specific diseases.
Yeah.
You mentioned a few earlier, but let's dive into the details from our sources. How does it play out in something like osteoarthritis?
Right, osteoarthritis, that debilitating joint disease where cartilage breaks down. Senescent chondrocytes, the main cells in cartilage, are major culprits. When they build up in the joint, they show high levels of senescence markers like P16-9NK4A. And the synovial fluid that lubricates the joint gets flooded with high levels of SSP factors, things like IL-1, IL-6, and MMP3, an enzyme that chews up cartilage.
So they're actively destroying the joint from within.
Pretty much. But the really promising news comes from animal studies. They've shown that senolytic drugs that clear out these senescent chondrocytes can remarkably rejuvenate the cartilage, even promote actual repair. That offers a truly transformative potential treatment.
Amazing. What about lung conditions, like pulmonary fibrosis?
Idiopathic pulmonary fibrosis, or IPF. It's a devastating progressive lung scarring disease with a really poor outlook. Senescent cells are deeply involved here too. You find senescent lung cells, alveolar type 2 cells, endothelial cells, fibroblasts contributing to the structural damage and scarring.
Oh.
These senescent cells express markers like P21, P16, and SA-gal, and the senescent fibroblasts, through their SAP, actively remodeled the lung tissue in a bad way, creating this pro-fibrotic, pro-senescent environment that just keeps the disease going. This is where xenomorphics, drugs that block SAP, are being explored. For instance, the antibiotic roxithromycin showed promise in lab studies by blocking a key SAP signal, TGF, potentially stopping further senescence and fibrosis in lung cells.
And the impact on our arteries,
So,
atherosclerosis.
in atherosclerosis, the hardening and narrowing of arteries, senescent cells in the blood vessel walls, both the endothelial cells lining the vessels and the smooth muscle cells within the walls, are major players in forming those dangerous plaques.
How do they contribute?
Senescent endothelial cells become leaky because of their SISP. They let immune cells and fats infiltrate the vessel wall, kicking off plaque formation. Senescent smooth muscle cells add to the fire with their own SAC, promoting inflammation and making the plaques unstable and more likely to rupture, which can cause heart attacks or strokes.
So can we target this?
Preclinical studies suggest yes. Senolytics have been shown to improve plaque stability and reduce plaque formation in animal models. And drugs with xenomorphic properties like rapamycin and ruxolitinib that hit that mTOR pathway involved in CSP production are also being intensely studied for atherosclerosis.
Alzheimer's, one of the most feared diseases of aging. Where does senescence fit in?
The link between Alzheimer's disease and senescence is looking increasingly profound. Senescent astrocytes, those star-shaped support cells in the brain, seem to actively contribute to forming both the amyloid beta plaques and the neurofibrillary tangles, which are the hallmarks of Alzheimer's.
They help form them.
Yes, and they also seem to impair the brain's immune cells, the microglia, from clearing away these toxic protein clumps effectively. The chronic SAP from senescent astrocytes might even directly cause tangle formation in nearby healthy neurons, spreading the damage.
A vicious cycle again.
It seems so,
Yeah.
and it might go both ways. The toxic amyloid proteins themselves might trigger astrocytes to become senescent in the first place. But importantly, studies using senolytics in mouse models of Alzheimer's have shown they can actually alleviate cognitive deficits, possibly by targeting other senescent brain cells involved in maintaining neuronal health.
It really feels like these zombie cells are everywhere, contributing to so many different problems. What about that general physical decline, the weakness and vulnerability we often associate with old age?
That brings us to frailty. Frailty isn't just being old, it's a specific clinical syndrome. It involves progressive physical decline, weaker muscles, sarcopenia, slower walking speed, general weakness, being much more vulnerable to stressors like infections or falls.
And senescence is linked to this.
Strongly linked. That systemic inflammation we talked about in inflammaging, driven primarily by the buildup of senescent cells and their SISP, seems to be a major cause of frailty. Pro-inflammatory factors like IL-6 and TNF-cell, pumped out by senescent cells, directly contribute to this chronic inflammatory state. The muscle loss, the weakness, even cognitive decline seen in frailty are increasingly being tied to senescent cells disrupting normal body functions. It really highlights the difference between just chronological age years lived and biological or physiological age, which reflects this deterioration. Senescence heavily influences biological age.
This really cements cellular senescence as a central player.
Right.
But wait, I remember hearing something that sounds like a contradiction. Doesn't senescence also play a good role? Specifically regarding cancer, how does that fit with everything we've just said?
Ah, yes. That is one of the most fascinating and sometimes confusing paradoxes in the whole field. Senescence has this crucial dual role when it comes to cancer.
Okay, explain the good side first.
On one hand, it's a powerful tumor suppressor. When a cell gets damaged that could potentially turn it cancerous, senescence kicks in and puts a permanent stop to its division. Cell cycle arrest. This prevents a potential tumor from ever forming or stops it very early.
Containing the threat.
Exactly. And in this scenario, the SASP can even be helpful initially, calling in the immune system to clear out those damaged premalignant cells. In fact, many standard cancer treatments, like chemotherapy and radiation, actually work partly by inducing senescence in the cancer cells, stopping them from multiplying.
Okay, so stopping cancer, good. What's the paradoxical bad side then?
The paradox comes with the long-term effects. While that initial senescence is protective, if those senescent cells hang around chronically, their persistent SISP can create a local environment, a supportive niche that actually helps existing tumors grow, spread, and even relapse after treatment.
Wow, a double-edged sword.
Absolutely. Which is why cancer therapies are complex. Some drugs, like palbociclib, work by inducing senescence in cancer cells to stop them dividing. But other research shows that senolytics, which eliminate senescent cells, can prevent cancer progression in mice and even boost the effectiveness of some chemotherapies. Researchers are even developing super-specific tools like CAR-T cells engineered to hunt down senescent cells based on unique markers, not just for cancer, but maybe for lung or liver fibrosis too. It just shows how nuanced the approach needs to be.
So depending on the timing and context, senescence can be a vital shield against cancer or, ironically, fuel for its fire.
Yeah.
That's incredibly complex.
It really highlights how context determines outcome in biology. And beyond these major examples, the impact of senescence is broad. It's implicated in chronic kidney problems, type 2 diabetes development, non-alcoholic fatty liver disease, NAFLD, the general decline in immune function with age, even cachexia, that severe wasting syndrome seen in chronic illness. The more we look, the more senescent cells seem like a pervasive driver of aging's downsides.
This incredible progress, understanding, and even starting to manipulate aging at this deep cellular level, it inevitably raises huge questions. So what does this all mean for us, for society? If we really gain the power to extend healthy life by targeting these cells, what are the ethics involved?
This stops being just science, right? It becomes deeply societal, philosophical.
You've hit the nail on the head. This is where the conversation gets truly profound, moving way beyond the lab bench. There's a really insightful paper by Tom Mackey from back in 2003 that laid out a framework for these ethical debates, and honestly, it's still incredibly relevant today.
Yeah.
It maps out the competing arguments we have to wrestle with.
Let's start with the arguments against pursuing anti-aging medicine widely. What are the biggest red flags, the main concerns?
The first, and maybe the most visceral concern, is inequity. There's a massive fear that these advanced, likely expensive technologies would only be available to the wealthy, at least initially.
Creating a huge gap. A potentially massive, unprecedented societal divide. The source puts it starkly. A future where the poor die young by the millions, while the rich refuse to age. That forces fundamental questions about justice, who gets access to a longer, healthier life. What does that do to society?
That is a chilling prospect. Exacerbating existing inequalities in such a fundamental way. What other objections are there? Philosophical ones, maybe?
There's a strong argument about denying aging's immutability. This view sees aging as just, natural, maybe even divinely intended, something humans shouldn't meddle with. Tied to that is the concern about dominating nature and altering ourselves. Worries about turning the human body into a commodity, about losing something fundamental about our identity if we drastically change our natural lifespan.
Hubris, essentially. Playing God.
That's often the underlying fear, yes.
Yeah.
And then there are very practical global worries.
Resources, right. That jumps out immediately if people live much longer.
Exactly. The specter of overpopulation. If lifespans are drastically extended, but we don't have equally radical solutions for food, water, energy, and don't fundamentally rethink our social and economic structures, it could put immense strain on the planet. Some even raise concerns about the rights of future people to be born if current generations essentially hog resources and opportunities for far longer.
A heavy burden on future generations. What about the individual experience? Living for, say, 150 healthy years? Could that have downsides?
That brings up the idea of ennui, profound existential boredom. Could a life with no natural deadline, as the paper puts it, eventually outlive its value? Would extreme longevity lead to meaninglessness, a loss of purpose? It's a deep philosophical question. And finally, there's the worry about ageism. It could cut both ways. Maybe increased prejudice against those who age naturally, either by choice or lack of access, or perhaps prejudice against the young, whose opportunities might feel blocked by an older generation that just doesn't move on from positions of power or influence.
These are definitely serious, complex issues. A challenging future to navigate if anti-aging tech becomes common. But Mackey's paper also laid out strong arguments for pursuing it, right? What's the case in favor?
Yes, absolutely. The foundational argument on the advocacy side is beneficence, the core ethical duty of medicine.
To help people reduce suffering.
Precisely. To maintain health, prevent disease, delay death. From this viewpoint, aging isn't just natural. It's the single biggest risk factor for almost all the chronic diseases that plague us: cancer, heart disease, dementia, diabetes. So medicine has a moral imperative to tackle aging itself as a treatable medical issue.
So it's an ethical obligation, just like fighting any other disease. What about the practical side? Public health economics.
That leads to the argument for efficiency. Proponents say, look, if we can slow down the fundamental aging process, we'd simultaneously slash the rates of all these major diseases. Instead of playing medical whack-a-mole, treating each disease separately as it pops up.
Which is expensive and often happens late.
Right. Targeting aging at its roots is potentially a far more efficient, holistic, and impactful public health strategy. Prevent the whole cascade of problems before they start.
Addressing the underlying vulnerability itself. Makes sense. What else?
There's the argument for limited autonomy. Basically, individual freedom. People should have the right to choose anti-aging treatments if they're proven safe and effective based on informed consent. It's about personal choice over one's own body and lifespan. And maybe the most appealing argument, the promise of improved quality of life. The goal isn't just more years, but more healthy, active, fulfilling years, a longer, more active, healthier, and fuller life. The idea is that wisdom and experience could keep growing instead of being cut short by decline. A future where later life means vitality, not just infirmity.
So after weighing all these pros and cons, the societal risks versus the potential benefits, what was the bottom line in Mackey's 2003 assessment?
The conclusion back then was that the arguments in favor of pursuing anti-aging medicine were ultimately found more compelling than the arguments against. It led to a recommendation for continued funding and appropriate regulation to responsibly develop this field. The vision was not just reducing disease, but enabling longer, fuller, more meaningful lives for more people.
It's an incredible balancing act, isn't it? Almost dizzying. How do we, as a society, navigate this? Balance the huge potential upsides, freedom from disease, longer opportunities, with these really serious concerns about equity, resources, even what it means to be human. It feels like a future rushing towards us when we're only just starting to grasp.
It absolutely underscores the need for ongoing, robust ethical discussion alongside the scientific progress. We have to ensure these revolutionary breakthroughs benefit humanity broadly, don't create new, deeper divides, and are accessible. That needs foresight, global cooperation, thoughtful policy.
Beyond just tackling senescent cells, what other big breakthroughs or promising areas in aging research are bubbling up right now? Our sources hinted at a wider landscape.
Oh, definitely. While senescence is a huge piece of the puzzle, the broader field of geroscience is incredibly rich. One area that's maybe less flashy but profoundly beneficial is simply exercise.
Good old exercise.
You bet. Research by scientists like Dr. Nathan Lebrasseur and Dr. Ozioma Okonkwo shows clear cellular and cognitive benefits. Regular activity protects against diabetes-like issues, offers neuroprotection against Alzheimer's, even for those at high genetic risk, and boosts overall cognitive function. It hits aging processes at multiple levels, from metabolism to inflammation. It's fundamental.
The simplest intervention is still a powerhouse. Okay, what about some of the more cutting-edge stuff? I think you mentioned young blood.
That's right. Work pioneered by Dr. Thomas Rando, among others, has shown fascinating things. The blood of young animals seems to contain specific factors that can actually rejuvenate organs and tissues in older animals. Heart, brain, muscle have all shown effects.
Rejuvenate. Seriously.
It appears so. They're still pinpointing the exact molecules responsible, but the implications for potentially delaying dementia, heart failure, muscle loss in humans are obviously significant. It's being actively pursued.
And diet, naturally, is huge. But it's evolving beyond just cutting calories, isn't it?
Absolutely. Dietary restriction research is much more nuanced now. You have researchers like Dr. Velter Longo developing fasting mimicking diets, short periods of significantly reduced calories that trigger beneficial metabolic shifts and lower disease risk factors.
Okay.
And then there's Dr. Satchidananda Panda's groundbreaking work on dietary timing. It's not just what you eat, but when. His research on time-restricted eating, limiting your eating window to maybe 8 or 12 hours a day, shows amazing benefits in mice. Reduced fat, less inflammation, reversal of type 2 diabetes and fatty liver disease, even increased endurance, even if the diet itself isn't perfectly healthy. Aligning eating with our circadian rhythms seems key.
That's fascinating and potentially very practical for people. What else is on the horizon, especially for things like Alzheimer's?
A huge focus is preventing protein aggregation. That buildup of misfolded proteins into toxic plaques and tangles is central to Alzheimer's and Parkinson's. Dr. Ana Maria Cuervo is a leader in autophagy research that's the cell's cleanup system for recycling waste, including damaged proteins. Her work shows autophagy failure is a major contributor to aging and related diseases.
So, boosting the cell's garbage disposal.
Essentially, yes. And others, like Dr. Andrew Dillon, are digging into why cells lose control over protein quality with age and how to fix that protein misfolding stress. New therapeutic targets are emerging there.
It feels like we're getting right down to the most fundamental breakdown processes.
Yeah.
What about actually reversing age at the cellular level? That sounds like pure sci-fi.
Well, it's moving towards science fact. This is cellular reprogramming, maybe one of the most mind-bending areas. It turns out if you briefly switch on just a few specific genes, often called Yamanaka factors, you can take almost any mature cell, skin, liver, whatever, in a lab dish, and revert it back to a youthful stem cell-like state. Dr. David Sinclair has done key work here, asking if aging is fundamentally driven by epigenetic changes, the instructions layered on top of DNA, and if we can reset that epigenome. Recent mouse studies are stunning. Briefly, carefully toggling these genes on and off in vivo seems to restore youthful function in muscles, metabolism, even damaged optic nerves in old mice. The potential for human rejuvenation, it's just staggering to think about.
Truly mind-blowing. Reversing biological age, not just slowing it. Are there other key biological differences being studied?
Yes, a growing realization is that biological sex differences matter in aging. How aging unfolds and how interventions work might differ significantly between males and females.
How so?
Research by Dr. Stephen Ostad and Dr. Dina Dubal looks at unique aspects of male versus female aging, even the role of the X chromosome in resilience. Dr. Dudley Lamming's work investigates why responses to things like rapamycin might differ based on SARM hormones or specific genes. It all points towards needing more personalized anti-aging approaches. What works best might depend on your biological sex, among other factors.
And how do we even track if any of these interventions are working in people? Is there a definitive biological age test now?
That's been a huge quest, but yes, there's been a major breakthrough. Reliable biomarkers of biological age. For years, scientists sought markers to tell us someone's true physiological age, distinct from their birthday number. The big discovery is epigenetic clocks.
Epigenetic clocks.
Yes, specific patterns of chemical tags, DNA methylation on our genome. These patterns turn out to be incredibly reliable indicators of biological age in pretty much all mammals, including us. Work by Dr. Morgan Levine and others has been key in validating these. This gives scientists powerful tools to objectively measure if an anti-aging therapy is actually making someone biologically younger. It's a game changer for tracking progress.
Okay, so we have these incredible revolutionary breakthroughs, but it's not all solved yet. What are the big lingering questions, the remaining complexities? Where does the field go next?
Right. It's crucial to acknowledge the complexities. A major one is the sheer heterogeneity of senescence. Not all senescent cells are identical. Their specific characteristics, even the exact mix of their SAC signals, can vary a lot depending on the tissue, what caused them to become senescent, the person's overall health.
So one size won't fit all for treatment.
Exactly. There's no single gold standard biomarker that flags every single senescent cell. And the SASP itself is dynamic, it changes. This complexity makes drug targeting really challenging.
And that dual nature, sometimes good, sometimes bad, that keeps coming up. How do you get rid of the bad ones without losing any good ones or disrupting vital functions?
That is the therapeutic puzzle. And there are specific potential hurdles with senolytic therapies, too. For instance, some animal studies show senescent cells can actually come back after treatment stops.
Uh-oh, so it might not be a one-and-done fix.
Maybe not. It suggests we need to understand the root causes better, maybe use pulse therapies or ongoing strategies. There's also a theoretical concern about potentially accelerating stem cell exhaustion if senolytics are used too aggressively over long periods, as senescent cells sometimes act as brakes on stem cell division.
And what happens to the dead zombie cells after senolytics kill them? Does our body clean them up okay?
That's another critical point. The immune system is supposed to clear the debris. But what if the patient is already old or sick with a weakened immune system? Could the cleanup process falter?
Leading to more problems.
There's a theoretical risk of something called secondary necrosis, where the dead cells break apart messily and release inflammatory signals themselves. That could actually worsen inflammation, undermining the therapy's goal. So ensuring the immune system can handle the cleanup, or maybe finding ways to boost it, will be important for widespread use, even if the benefits currently seem to vastly outweigh these potential risks.
So, bottom line, continued, deep, nuanced research is absolutely vital.
Absolutely. Relentless, granular research is needed to fully map this complex cellular state and its impact. But the overall message, strongly backed by the evidence we've discussed, is that aging and age-related diseases are potentially modifiable, maybe even curable or significantly delayable through human intervention. But it demands a sophisticated, context-aware approach. The future is likely precision aging medicine.
Oh, what an incredible journey today. From those microscopic zombie cells hiding in our tissues, all the way up to these huge, profound ethical questions about extending healthy life. Understanding aging at this deep cellular level, it's genuinely transforming medicine, pushing boundaries we once thought were fixed.
It really is. The breakthroughs in just detecting these cells, electrical signatures, non-invasive MRI, and then the therapies aimed at them, the senolytics, the xenomorphics, it's truly profound. It points towards a future where maybe we don't just manage the symptoms of aging, but actually prevent or even reverse the underlying processes.
It leaves you thinking, doesn't it? What stands out to you from all this? If we really do manage to extend health span, not just add years, but add good, vibrant years, think about the ripple effects. What new opportunities open up? For learning, for careers, for just living? How might families change? And maybe the biggest question, how does our whole definition of old evolve?
It's a future that's moving from science fiction to potential reality faster than many people realize, and we're only just starting to grapple with what it all means.
Thank you so much for joining us for this deep dive into the future of aging. It's been truly fascinating. We'll be back soon with another deep dive into the topics shaping our world.
Thanks for listening. For all things health and fitness, subscribe to our newsletter today. Find out more at armygymnastics.com.